Hello, everybody. How are you? Here we are again with a new topic which talks about application of biotechnology. Biotechnology in industrial biotechnology. Simply, it does mean. Therefore, today we shall talk about briefly, just what industrial biotechnology means. Simply, it says application in industry. Um, Industrial production of enzymes is one area of the white. This industrial biotechnology, by the way, is color coded by white. The white biotechnology in one area of white biotechnology or application of biotechnology industry is a production of enzymes. Enzyme production or enzymology. Applied enzymology is a very huge and lucrative business. Of course, enzymes are produced for various purposes, such as to be used as detergents or to be used in food industry to process different chemical reactions. Of course, therefore they are needed in many areas, in many industries as raw materials for biochemical processing or reactions. Therefore, it is huge industry, well-developed industry, and lucrative. Of course, it's profitable industry. Of course, therefore, let's see industrial biotechnology in terms of enzyme production. Enzymes are a group of globular proteins, compare it from fibrous or structural proteins. These are globular, three-dimensional spherical proteins. Globular proteins that serve as biocatalysts. That means catalyze biochemical reactions. Just they help to accelerate or speed up reactions, the rates of reactions, of course. Industrial enzyme production involves the use of microorganisms to produce enzymes that are utilized across various industries, including food industry, pharmaceuticals, and biofuel industry, blah blah blah. Therefore, detergent industry even. Therefore, industrial enzyme production involves the use of microorganisms, fungi, and bacteria, and other microbes to produce enzymes that are utilized across various uh, other factories or industries like detergent industries, like food industry, like pharmaceuticals industry, or biofuel industry. Then enzymes are produced commercially by using readily available feedstock. You see, whenever we are thinking of uh, production of a certain substance by using microorganisms, we have to provide substrate. Substrate. What is the source of the substrate? The raw material on which the microorganisms act and give us the product that we need, either enzyme or any pharmaceutical chemical or drug or antibiotic, whatever. Therefore, enzymes are produced commercially by using readily available feedstocks. What are these feedstocks or the substrates? The raw materials that provide organic nutrients for the microbes to produce the chemical of our desire. One, um, corn steep liquor, sugar containing fluid, primarily made from corn flour, of course. The other is molasses. This is a byproduct of the sugar industries. Uh, Agro-industrial wastes from the agro-industry, like fruits, rotten fruits or spoiled fruits, smashed fruits, uh, and other agricultural wastes, plant sources, plant cellulose, lignin, if you like, hemicellulose. All this can be used as raw material substrate for growth of microbes so that they can produce the industrial product they need. We need enzymes. Yeah. Fungi, aspergillus, and bacteria, bacillus are the common organisms used in enzyme production since they are non-pathogenic. You see, when we are planning to produce a certain chemical by using microbes at industrial scale, one criteria we need from the microorganism that we use is that microorganism should not be pathogenic. If pathogenic, working with this pathogenic microbe in order to extract something beneficial is uh, has its own risks. There may be contamination, so that if it is a serious pathogen, this may be spread in that community, so that it can lead in some ailment, in some health problem. Therefore, one criteria is fungi, aspergillus, from fungi, aspergillus, and from bacteria, bacillus. Bacillus is this genus of bacteria is also known in antibiotic production. It's really smart. Bacillus is also known in insecticide production. It's from agricultural biotechnology, or agricultural biotechnology lecture. Therefore, these are really smart, super smart bacteria, utilized in many areas. And therefore, here we use this bacteria to generate the enzyme that we desire, of course. And therefore, the other, if the enzymes produced are extracellular, the liquid feedstock, you see, if the enzyme that the microbes produced inside and released outside. If the enzymes are produced and released out, they are known as extracellular. The liquid feedstock is filtered from the microorganism. When we finally separate, we filter the liquid from the microorganisms. There is a filter which filters the microorganisms separating from the remaining liquid. And from the remaining liquid, we extract the enzymes. You see? And the enzymes are filtered out from the liquid feedstock then after. If the enzymes are intracellular, you know that if they are produced inside the cells of the microbes, and if they are not getting out, just produced inside and remain inside, we have to break the cell or the microbes by different mechanisms. So vibration, from also by many laboratory techniques, the microorganisms have to be filtered from the first. We filter the microorganisms first from the feedstock, then the microorganisms are crushed or broken open to release the enzymes. The enzymes are then taken up or separated from the other part of the microorganism. You see, in intracellular enzymes, we have to kill the microbes. Not only killing, we have to smash open or break them open, so that released from the cytoplasm, the enzyme is released. Um, this is the plant that we use for enzyme production. It's similar with that of antibiotic production, the fermenter or the bioreactor. It has a steering paddle. The contents of the fermenter, the substrate plus the microorganism must be moved or stirred. And suitable conditions must be maintained, that is suitable temperature and suitable pH. And finally, we have to also supply air. It's aerobic process. Also, the fermenter must have the water jacket, of course, whenever there is high heat production inside and it becomes hot, and excess temperature can denature the enzymes of the microbes and kill, so that production stops. So we have to maintain a constant temperature by running cold water when it becomes hot. And then the diagram to the left is at the end of the fermentation, the reaction process. Material, the enzymes are separated by the process generally known as the downstream process, downstream processing, filtration, separation of the enzyme, then purification, and then we may prepare it in different forms as a liquid or as a crystal, whatever it is. Therefore, downstream processing involves all this until packaging the product, purifying, concentrating, and even checking whether it is pure enzyme or whatever. We have to check first before packing. Therefore, similar to the antibiotic production system. Uh, the knowledge of genetic engineering also used for large scale production of enzyme by using microorganisms. Yeah, we can insert. We have to prefer when, if we get certain enzyme that we desire, we need from a microorganism, very essential enzyme. But if that microorganism from which we are going to uh, uh, by which we are going to produce enzyme is not uh, good to be reproduced in the lab condition. Uh, slow growth rate. I mean, larger generation time, requirement of specialized growth factors and nutrients. That is not profitable. So that we can take the gene and insert that gene into other bacteria such as E. coli, uh, which are really very good in fast growth rate and or short generation time. Plus, they require less expensive nutrition to grow in controlled condition. Therefore, the knowledge of genetic engineering is also used for large-scale production of enzyme by using microorganisms. And the other today, enzyme production technology is one fast-growing, as I've told you, fast-growing technology. Enzymes are used for an increasing range of applications, uh, various different industries: the food industry, pharmaceutical industry, and the energy industry, energy production, biogas production. In pharmaceuticals, enzymes are used in detergent industries as cleaning agents. Now, shall we see some commercial use of enzymes. Uh, Most common commercially produced enzymes are proteases, protein breaking down enzymes. This is group name. It's not a single protein, of course. Proteases. All those enzymes in this category can break down at different ways, different levels, at different specificity. They break down proteins, and the others are lipases. These also break down different sorts of lipids. Therefore, they are very much important in food and textile industry. These two. Enzymes are biological washing powders. This is one area of application of enzymes. Enzymes as biological washing powders or liquids, it could be. Biological washing powders or biological detergents contain a mixture of enzymes, including proteases and lipases, especially. Of course, some biological detergents may contain also amylases. Even some may contain cellulases if we are going to use these enzymes in textile industry in order to remove cellulose fibers from our clothing on the surface of clothes, and so that the clothes will have a smooth shiny appearance. Therefore, uh, proteases remove stains of proteins such as egg yolk. If there is a stain of protein, egg white or egg yolk or blood stain. Proteases, amylases in dish washer detergents, they are together used to wash dishes, industrial dishes. Uh, Lipases are lipid dissolving enzymes that are obtained from Candida lipolytica and Geotrichum candidum. You see, these are the two: Candida lipolytica and Geotrichum candidum. These are fungi, of course, fungi that are used to produce lipases. And lipases are used in washing powders for removal of fatty stains on clothes or other materials. If your clothes comes in contact with oil or any fat stain, it can easily remove efficiently as washing powder. Lipases are also used to enhance flavor in cheese. We add different types of lipases in cheese production so that these lipases bring about some desired flavor to your cheese, liked flavor, good flavor, so that marketable flavor, of course. Um, Dentures can be cleaned with products containing a protease to give a better result in shorter time. Dentures are also cleaned by using enzymes. The other contact lenses are also cleaned efficiently by using enzyme combination, either proteases or lipases, or combination of these two. Contact lens cleaning. Pectinases, one important area of application of enzymes is pectinases. These enzymes are very crucial in the fruit industry, fruit production, fruit juice production, of course. Pectinases are obtained commercially from Bisoclamis fulva. Bisoclamis fulva. They are used in clarification of fruit juice and also maximize the volume of the fruit juice. You see, when juice is produced, juice contains normally fibers known as pectin fibers. The presence of pectin fibers from the juice gives a cloudy appearance to the juice, which is not attractive for the sight. So if we apply pectinases, these substances, polysaccharides known as pectins, will be broken down into simpler sugars. This has multiple purpose. One, it removes the cloudy appearance so that a clear natural color of the fruit is seen. Two, it also adds volume. The volume of the juice is also added. Plus, it also enhances its taste, the sweetness, and enhances its taste. These are the three advantages of using pectinases as to clarify fruit, not only to clarify, of course, primarily we wanted to clarify the cloudy appearance of the fruit so that it will be bright yellow, bright red, bright orange, whatever color. Cloudiness is removed and also volume is increased and also taste is also enhanced, sweetness. Triple benefit, of course. Streptokinase, or in other words, tissue plasminogen activator, is an enzyme. We have seen this in human health and well-being, of course, section. Is an enzyme obtained from the cultures of some hemolytic streptococci. Streptococci are the genus of bacteria that contain species that produce streptokinase, of course. And what is the purpose of this streptokinase? This break down blood clots in the circulatory system. It has fibrinolytic effect. That means breaking down the fibers that form blood clot, and therefore it helps in clearing blood clots inside the blood vessels through dissolution of intravascular fibrin. It dissolves intravascular fibrin. Fibrins are responsible for formation of blood clots, and if you break down the fibrins, the blood clot will be dissolved so that no block. If blood vessels are blocked, blood flow to the tissues after that blockage area will be decreased or totally stopped, which means those tissues become necrotic because of absence of oxygen. They will start to die. Imagine if this death is in the heart muscle itself, heart attack. If it is in your brain, that means that part of your brain is not functioning. Therefore, it's a big problem. The other purpose of enzymes is meat tenderizing enzyme. Meat tenderizing. Therefore, we have meat tenderizing enzyme. Tenderizing means making soft, chewable. Therefore, meat tenderizing means to soften meat for cooking, and the two most often used meat tenderizing enzymes are papain, obtained from papaya, it is a protease, bromelia, another protease obtained from pineapple, from the plant source pineapple. Therefore, you can see these are very important industrially applied or used. And other industrial products we have seen so far briefly. One big industrial product, the white industry product, that is enzymes. Briefly, you have seen. The other industrial products may be these products are organic acids. Organic acids are just similar to enzymes. Organic acids are also needed in many different industries, starting from pharmaceuticals to the dye industry, textile industry, whatever. And microbes are also used for commercial industrial production of certain organic acids such as acetic acid, citric acid, and butyric acid, propanolic acid. whatever propionic acid include acetic acid. There are mainly two steps in acetic acid or vinegar, the other name for acetic acid vinegar production. One, the first stage, yeast converts sugar into ethanol by anaerobic means anaerobic respiration. While in the second step, ethanol is oxidized to acetic acid aerobically, the presence of air, I mean oxygen, by Acetobacter aceti. This is the renowned bacteria that converts ethanol into acetic acid. This process is called acidification, the second process. The first process is ethanol fermentation. Of course, it is done by yeast. Therefore, yeast then Acetobacter aceti finally give us what? The acetic acid. The product is acetic acid, organic acid. And of course, this organic acid is employed in pharmaceuticals, coloring agents, insecticide, plastify, many different other factories. Of course, they are very important raw materials. The other acid is citric acid. Citric acid is obtained through the fermentation carried out by Aspergillus niger, its fungus, and Micor, also another fungus. Or when they act on sugary syrup, a syrup or a fluid which is full of sugar. You can make this sugary syrup from cereals like corn. Citric acid is employed in dyeing industry, engraving, medicine, inks, flavoring, and preservation of food and candies. Of course, we apply it to food to keep our food from spoilage, of course. The other is lactic acid. Lactic acid fermentation is carried out by both bacteria, Streptococcus lactis, Lactobacillus species, and also fungi like Rhizopus. Lactic acid also is important as industrial raw material in many industries. The other is butyric acid. Butyric acid is made by Clostridium acetobutyrica. It's a bacteria that is used for production of butyric acid. You see, this also has many industrial application. In many industries, it is used as raw material in dyeing, in pharmaceuticals. Many industries use organic acids, and even in cosmetic industry. And this is about industrial application of biotechnology, the white biotechnology. And